Well, thank you for coming to the lunchtime spruce talk. My name is Matthew Renner, and I was humbled and a little bit embarrassed to receive the 2019 Richard Spruce Award. The least I can do in return, of course, is give a talk, and here we are. Unfortunately, I haven't been doing much bryophyte research for the last couple of years, so what I have to present is not quite new. It's based on a publication from last year, onto which I thought I would tack some unlikely extrapolations from the improbable architecture of Herzogianthus vaginatus in a presentation that is co-authored with Joshua Salter, who's an independent researcher in Auckland. And of course I'm a research scientist based at the Royal Botanic Gardens in Sydney. Now, Herzogianthus is a genus of one, maybe two species. It's endemic to New Zealand and is known from 9 sites in the North Island and 3 sites in the South Island. It was recently placed into the family Herzogianthaceae, which is probably monotypic. The photograph on the left shows what it looks like in the field. You get a sense of the plant's architectural complexity when you encounter it in the field. And on the right you see a shoot in close-up. Now, this impression of architectural complexity is only enhanced when you look at it under glass. It's a striking and beautiful liverwort with regularly lobed, succubusly inserted leaves in which the dorsal lobe is swept a long way forward. They're covered in long cilia. The shoot systems are regularly pinnately branched by terminal Frelania type branching. The secondary shoots have vaginate leaves and are often determinate in length. They get to a certain length and then stop. There's of course a regular left-right alternation of leaves on the dorsal surface. And the first interesting feature, in addition to all the others, the first interesting feature that you come across in Herzogianthus is that the underleaves are not always regularly spaced. You can see that in the image on the right, where the lowermost 4 underleaves have a big gap between the second and third. If we take a close look at the secondary shoots of Herzogianthus, we see another interesting feature, which is that the leaves are very crowded. Now, if we count the number of leaves on one side of the lower secondary shoot, there are 12. So there's 12 pairs of leaves. But interestingly, there's only 6 underleaves, which is around half the number that there should be if the shoot grew by a regular helical segmentation sequence. This is very, very strange, and it's true of all secondary shoots. There's only around half the number of underleaves there should be. Another unusual feature occurs on the primary shoots, and it's associated with the leaf and underleaf sequence. Now, in shoots that grow by regular helical segmentation, the leaf, or the branch if it's terminal, preceding sequential underleaves is always on the same side. But you can see here in Herzogianthus that the lateral appendage subtending the lower underleaf, which comprises a half leaf and a branch, is on the left, while at the upper underleaf, the lateral appendage subtending the branch, which again comprises a half leaf and a branch, is on the right side of the shoot. And further, even though the branch at the base of the upper underleaf is inserted below that underleaf, i.e. between it and the previous underleaf, there are also another 2 lateral appendages, this time leaves. They're labelled SL, stem leaves, separating these sequential underleaves, which are labelled SU, stem underleaves. So the fact that there are 3 lateral appendages separating 2 sequential underleaves is very unusual. It shouldn't really happen. And the first question then is whether the separation of sequential underleaves by more than 2 leaves is a common occurrence within Herzogianthus. To address that question, Josh and I took 3 specimens from the Mangawhero Falls population of Herzogianthus vaginatus and counted the number of lateral appendages separating 295 sequential underleaves on primary shoots and 239 sequential underleaves on the 69 secondary shoots attached to them. And we plotted the data. And you can see that there's some interesting features in this data. On primary shoots, the underleaves, sequential underleaves, were most frequently separated by 3 lateral leaves or half-leaf branch pairs. Sometimes, though, they were separated by 4 lateral leaves and sometimes 2. In 3 instances, sequential underleaves were separated by only 1 lateral leaf. On secondary shoots, a slightly different pattern emerged. Most underleaves were separated by 4 leaves, sometimes, though, by 3 and rarely by 2. Another interesting feature is that when you take the shoots as a whole, the shoot mean leaf to underleaf ratio is more variable in secondary shoots than in primary shoots, and further, that ratio in secondary shoots exhibits a significant relationship with shoot length, in that longer secondary shoots have higher leaf to underleaf ratios. So, in summary, Herzogianthus systems possess 5 very interesting features. We're going to go into these interesting features in some depth. None of those 5 interesting features is compatible with the 2 segmentation sequences known in leafy liverworts. Now, for clarity, my use of the term segmentation sequence refers to the sequence of cell divisions that occurs at the apical meristem. These divisions result in derivatives called merophytes, and from each merophyte, by subsequent cell division, leaf and stem tissue comprising shoots are produced. Usually all 3 merophytes contribute to stem tissue, and they also produce leaves and underleaves if you're the ventral merophyte. Now, segmentation diagrams, such as the 2 presented here, have been used to illustrate segmentation sequences and concepts such as anodic, cathodic, polarity in relation to helication direction since at least Lightgib in 1871. They've also been used by others, including Evans, Crandall-Stotler and Schuster. They're very useful diagrams. You can get a lot of information from them, and that's that partly explains their popularity. These 2 segmentation diagrams represent the only known growth patterns in leafy liverworts. Helical on the left, where divisions from the apical cell occur in a regular cyclical series, and pendular on the right, where divisions occur in a pendular sequence, which produces as many leaves as underleaves. Now, just out of interest, pendular segmentation is a feature of early spalling growth in Frillaniaceae and Leguniaceae before helical segmentation commences in mature spalling. In a couple of genera, including Cola legunia and Diplasia legunia, the shoots retain this juvenile growth pattern and never grow by helical segmentation, except within male branches. So, the expression of sexual structures in those genera is linked with the expression of an adult growth pattern. And within single shoots, transitions to and from pendulous and helical segmentation occur. If anyone's aware of where that has been reported in the literature, I would appreciate being made aware of it, please. I'd be very surprised if the early morphologists have overlooked this, but I am not fluent in German, and I find that early literature very difficult to read. But I digress. Segmentation diagrams are useful devices. They capture positional information about segment arrangement and sequence, as well as leaf to underleaf ratios, and explain these in terms of a hypothetical segmentation sequence. So they're a valuable tool with which to approach understanding Herzogianthus shoot architecture. Now, as I said, most, and that's more than 90% of liverworts, grow by a helical segmentation, including this Dendrolimbidium tenax. It's important to emphasise that the regular helical segmentation is directly linked to the regular arrangement of leaves and underleaves on leafy liverwort shoots. So, to explain the unusual features of Herzogianthus shoot morphology, we might begin by imagining how those unusual features could be produced by shoots that grow via regular helical segmentation, given this is the most common segmentation sequence in leafy liverworts. Now, one possibility is that lateral merophytes are duplicated, and for this suggestion we thank Dr. Barbara Crandall-Stotler, who provided 2 excellent, challenging reviews of our early submissions on Herzogianthus. If alternate merophytes on primary shoots duplicated early in their ontogeny, and each duplicate went on to produce leaves and stem, this would result in a 3:1 leaf to underleaf ratio, as occurs on primary shoots. For secondary shoots, if every lateral merophyte duplicated, then a 4:1 leaf to underleaf ratio would result. Now, lateral merophyte duplication is not known in any liverwort, so this mechanism would be unique to Herzogianthus. Now, there are other possibilities as well, and we can use segmentation diagrams to model these alternatives to make predictions about what shoots growing by different processes would look like. Here we've got segmentation diagrams representing a range of modifications to merophytes produced by modifications to the sequence of merophytes produced by regular helical segmentation at the meristematic apical cell. In A, we have a segmentation diagram that is based on growth where the expression of every second underleaf is skipped, and this results in a 4:1 leaf to underleaf ratio. We also have the 2 merophyte duplication models discussed on the previous slide in C and D, and we have another duplication model where every second lateral merophyte duplicates rather than every alternate merophyte. And you can see the utility of segmentation diagrams here, because what at first seemed a reasonable thought, that every second lateral merophyte duplicated, results in a rather unbalanced and indeed unrealistic shoot morphology that has twice as many leaves on one side as the other, even though it predicts a leaf to underleaf ratio of 3:1. So here are 4 possibilities based on a regular helical segmentation sequence. Now, there are other possibilities if we permit departures from a regular helical segmentation. And now things start to get interesting. If we allow ourselves to permit the possibility that growth in Herzogianthus could depart from regular helical segmentation, then there are a range of ways in which such departures might occur. For our purposes, we'll focus on some rule-based departures where a change in the direction of helication, or a reversal, as indicated in grey, occur periodically, or at least at regular intervals. We've already seen one growth pattern that conforms to those requirements, which was the pendulous segmentation sequence occurring in Cola-Lejeune and Diplasia-Lejeune. That growth pattern can be thought of as being fundamentally helical, but wherein the direction of helication reverses after every lateral leaf, resulting in a pendulous sequence. So we use that as our starting point. If the direction of segmentation in Cola-Lejeune reverses every leaf, what about one where reversals occurred every second leaf? And it seems reasonable, and to an extent it is. But as you can see in D, in D above the top, it's the top left. This model predicts an unusual sequence of leaves and underleaves, where sequential underleaves are separated by either 1 or 3 lateral leaves in a repeated sequence. Now you get that from the segmentation diagram by looking at the number of lateral leaves that stack upon each of the merophytes representing each of the ventral merophytes. And you can see the very bottom one has 3 laterals stacked upon it, whereas the second from bottom has only 1 lateral stacked upon it. So this is the sort of useful information that you can glean from these segmentation diagrams. We might move from a model where reversals occur at every second lateral merophyte to a model where reversals occur at every third lateral merophyte, which is illustrated in E. This is unusual too, because it predicts a ratio of leaves to underleaves of 1.5 to 1. And we can move further to a model where reversals occur every fourth lateral merophyte. This predicts a ratio of leaves to underleaves of 2 to 1. And an unusual sequence wherein sequential underleaves are separated by a repeated sequence of 3, 2, then 1 lateral leaves. Trying reversals at every underleaf results in unusual patterns at the dorsal stem midline associated with sequential leaves on the same side of the stem. And we can try reversals based on other rules, for example, after 2 consecutive leaves, 3 consecutive leaves, and so on. We can also add reversals in sequence, so 2 reversals after 2 consecutive leaves. And 2 of those are illustrated in A and B, bottom middle and bottom right. Now, from those segmentation diagrams, which graphically represent each growth model, we can derive predictions about the shoot structure that each growth model would produce in terms of the leaf to underleaf ratio, the number of leaves separating sequential underleaves, the leaf sequence at the dorsal stem midline, the architectural sequence that repeats within each shoot, and the leaf preceding sequential underleaves, with which side of the shoot it's on. And this table summarizes all of those predictions about structure, the structure of shoots produced by each growth model. And with these predictions, we can compare the hypothetical growth models with the sequence in Herzogianthus primary shoots. We do this by translating the sequence in leaves and underleaves on Herzogianthus into a segmentation diagram via an intermediate step, which is a shoot map. And here we have, on the right, the shoot, the map, and the resulting segmentation diagram for one of the 15 shoots we mapped. In the segmentation diagram, we shaded every merophyte at which the next merophyte was out of sequence grey, and those merophytes are associated with a departure from normal helical segmentation. In other words, a reversal, just as in our hypothetical growth models. Having reconstructed segmentation diagrams derived from Herzogianthus primary shoots, we can compare them with the hypothetical models. The observed segmentation diagrams are on the left and the hypothetical models are on the right. We didn't do this for secondary shoots because the sequence of leaves and underleaves was too difficult to establish due to the crowding of leaves and their fusion, which obscures stem insertion lines. So, the first point here is that each of the Herzogianthus segmentation diagrams contained leaves and underleaves that were out of sequence with normal helical segmentation. In other words, we can infer many reversals in the direction of helication. The second point is that the occurrence of reversals are variable to some extent and more variable in some shoots than others. Underlying this variability, though, was a pattern of reversals after the production of 2 consecutive leaves, in particular in shoot E. The third point is that 3 reversals were associated with underleaves and resulted in sequential underleaves being separated by only 1 lateral leaf, not a pair of leaves. Now, on the basis of the criteria that we outlined regarding shoot structure under each of these hypothetical growth models, the primary shoots of Herzogianthus are compatible with only 2 of them, being hypothetical model B and hypothetical model D, because the primary shoot ratio is usually 3:1 and the sequence of leaves and underleaves is what is predicted by both those models. Now, there's problems with applying model D to Herzogianthus that we will come to shortly. So, as I've just said, 2 models may explain primary shoot morphology. The 2 models may also explain secondary shoot morphology, which are the consecutive reversals after consecutive lateral merophytes. That results in a 4:1 leaf to underleaf ratio or duplication of every lateral merophyte. But I have to stress that no model explains Herzogianthus vaginatus primary or secondary shoot morphology perfectly well, because there are some significant problems. The first is the observation of 3 instances where a single leaf separated consecutive underleaves. And the second problem is variation in the number of leaves separating consecutive underleaves on primary and secondary shoots. And we also have an unexpected observation to deal with. Now, before we get to the unexpected observation, I should point out that I think the observation of underleaves separated by 1 leaf, only 1 and not 2, is a critical falsification of the merophyte duplication model, because even without duplication, at least 2 leaves should separate every underleaf if the underlying divisions, the underlying growth at the apical meristematic cell is helical. It's not necessarily a falsification of a reversing model if we permit some variation in when and where reversals occur, as our count data suggest we should. Okay. And now the observation supporting a reversing model, which was unexpected, comes from underleaf asymmetry, which, when the underleaves are trifid, alternates up the shoot in concert with an imputed cathodic and anodic polarity associated with segmentation sequence reversals. In other words, a reversing model of segmentation potentially explains this underleaf symmetry as well as the leaf and underleaf sequence. And it does that because underleaf asymmetry is known in many liverworts, including the Lepidosiaceae. That asymmetry is consistent within shoots, but may differ among shoots due to the syntros or antros helication, which is established by the sequence of divisions within the merophyte derivative that produces the Frelania-type branch when it's formed. So, fluctuating asymmetry in underleaves is, in liverworts, directly tied to the direction of helication. And if we've got alternating asymmetry within a shoot, we might explain that in terms of alternating direction of helication. So, in summary, Herzogianthus is unique because its primary shoot architecture does not occur in any other leafy liverwort. It's even more unique because its primary and secondary shoots have different architectures, neither of which occurs in any other liverwort. And the growth processes producing these architecture, whether merophyte duplication or helication reversals, are therefore unique to Herzogianthus. Now, you've probably got the impression already that my preferred model to explain this unusual architecture is growth by periodic reversals in the direction of helication, because this can also explain unexpected observations of underleaf asymmetry, as well as the sequence of leaves and underleaves, and the fact that the lateral leaves alternate and interlock on the dorsal shoot surface. But whether this is the explanation remains to be confirmed by careful, targeted histological work. And I emphasize targeted because sometimes Herzogianthus primary shoots do grow by helical segmentation, or at least appear to. So you have to be very careful about where you place your sectioning work in order to capture reversals. So, given the direction of helication is reversing in the growth of Herzogianthus, we might explore some possible explanations. And the first possibility is that Herzogianthus possesses a mutation in the gene regulatory network governing division plane establishment and maintenance in its apical meristematic cell. Division plane establishments are a critical component of plant cell division. It's regulated by a complex network of genes from different gene families, and perhaps Herzogianthus possesses a mutation in one of these genes, and it may mean we can learn about the function of that mutant gene from Herzogianthus if we can identify it. And that concludes the sensible part of this talk. Now, with the second possibility, I'd like to take this opportunity to release some of my wilder speculations about Herzogianthus, simply because the most sensible explanation is not always the most fun. And possibility 2 is that there's incomplete coordination within the gene regulatory network governing cell division plane establishment and maintenance in the Herzogianthus apical meristematic cell. Because, and I say that because we know the shape of the apical meristematic cell varies across the liverwort phylogeny. Within leafy liverworts, the apical meristematic cell is tetrahedral with 3 cutting faces. In the sister lineage to the Jungermanniidae, the Metzgeriidae, the apical cell is lenticular and only has 2 cutting faces. I don't think there's been a study of apical meristematic cell shape evolution yet, but it would be fascinating because of the diversity expressed within the next sister group to the Jungermanniidae plus Metzgeriidae, the Papaliidae, with species in that lineage having lenticular, tetrahedral and cuneate apical meristematic cells. This makes intuitive ancestral state reconstructions almost impossible. For example, you can't even begin to guess what the ancestral state for the Jungermanniidae plus the Metzgeriidae might have been. It's certainly unknown. But what if the most recent common ancestor of the Metzgeriidae plus the Jungermanniidae had a lenticular apical meristematic cell, and the tetrahedral apical meristematic cell of Jungermanniidae was acquired along the Jungermanniidae stem lineage? This would involve a change in the apical meristematic cell shape, and perhaps also in the gene regulatory networks governing the establishment and maintenance of cell division planes. What if then Herzogianthus represented an intermediate stage in the incorporation of a new merophyte row into leafy liverwort form, one wherein gene expression and regulatory networks had not yet fully coordinated within the apical meristematic cell, with the result that there's sometimes expression crossover and perturbation in the establishment of division planes? Maybe. But maybe phylogeny reconstruction based on target sequence captures already falsified this hypothesis by resolving Herzogianthus among the leafy liverworts with the rest of the Tilidiales. Such a result would then imply a rescue hypothesis for this favoured idea of mine, in the form of two different modes of gene regulation to achieve regular helical growth, one each in the parallels and the Ungermanniales, and others in the remaining Tilidiales if these also express regular helical segmentation. Such might not be beyond the realms of possibility, given differences in gene regulation between given that differences in gene regulation between the parallels and Ungermanniales may explain the complete inability of the former to branch from its ventral merophyte. So, if the ventral merophyte, and in particular its underleaves, are uniquely derived within the Ungermanniidae, then underleaves are a synapomorphia for that lineage. This hypothesis has several ramifications. The ancestral leafy liverwort was not erect and isophilous, likely it was disticus. The capacity to branch from the ventral merophyte is of derived character, not a primitive one, and underleaves are unique to leafy liverworts, which means we can stop trying to see them in other lineages, including this one, Haplometrium. Having relieved our perception from the expectation that all leafy liverworts have underleaves, we can stop seeing underleaves where they do not occur and start seeing Haplometrium for what it appears to be, which is an erect isophilous liverwort with loosely spiral phyllotaxis whose divergence angle departs slightly from 120 degrees. Anyway, I promised this talk would explore some unlikely ideas from an improbable footing, and I hope the wild segue at the end here was a bonus. My intention in their sharing is to encourage us all to prepare for surprising, uncomfortable, and even confusing observational data, especially when we synthesize broad-scale studies of the evolution of form in bryophytes with the equally broad phylogenetic frameworks we are currently generating. And that's the end of the talk. Thank you for listening. I would have many people I would like to thank for their support during this study, in particular the organisers of the 29th John Child Bryophyte Workshop at Uakone for creating an opportunity to make collections of Herzogianthus, Ngāti Rangi and the Department of Conservation for joint permission to make those collections, Ewan Cameron and Tahara Rangatunga at the Auckland War Memorial Museum for their assistance with the resulting specimens, Barbara Crandall-Stotler and an anonymous reviewer for their thought-provoking comments and suggestions for improving earlier versions of this study, Sue Lindsay, while microscopy and microanalysis laboratory manager at the Australian Museum for facilitating the scanning electron microscopy examination, Jorn Henschel for a loan of Tilidiaceae specimens for comparison, and John Braggins for permission to use his photos of Haplometrium gibbsi. Thank you all very much, and thank you to everybody who has listened to this talk as well. It's been fun for me to share my ideas with you all.